Hello, people. Camera's still not working, right? That's okay. Celine, Ariana, Matt Bernhardt. Good to see you, Matt. Noemi Alonso. Bisuit. All right. Time is at 9:32. A little bit early. I mean, a little bit late, but not too many people here. Dante's here. Yay, Dante. Hey there, Beeswit. You're looking awake today. Yay. 1, 2, 3, 4, 5, 6. Let's have your faces up today because we're going to be doing a lot of talking to each other, I think, I hope. I've got one thing to talk about, and then I want to do some review for the exam that is on Thursday. Bum, ba, bum. And the thing I'm talking about, let's see, Noemi, Mike, Bezuit, Celine, Dante. I think you've all been in my drugs class, I think, right? So it's going to be something we've talked about already in the pharmacology of illicit drugs class, but it's important to get it down here because it'll be important in this class too. So, and I know, as I've said before, a lot of times you guys forget things from one class to the next. So, my God, my camera is horrible. I'm like the ultimate white guy. Look at this. I'm so white, I'm almost disappearing. Okay, don't hold it against me. All right, let's go talk about receptor downregulation, which again, I know you've heard about before, but it's a good review here. Cool picture, Anna, not mine. Okay, so we talked about G protein coupled receptors, and here you've got these sites for phosphorylation. These sites here, the red ones are for GRK that we'll be talking about today. These stars are for protein kinase A. Remember, each talk about in this class, I don't know what the hell I talk about. I got 3 classes this semester. I don't know what the hell I'm talking about. But anyway, there are different consensus regions that each kinase will phosphorylate. The protein kinase A is like KR blank S or T, or KR blank blank S or T. And then there's a little, the site is a little bit different here for GRK, a little more specific, but I don't remember it. You know, do you ever get like, when you get close to a test, it's like, I just don't, just get it on. Anyway, receptor downregulation. So let's see, is this all I have? Nope. Okay. So here's your G protein coupled receptor here. So what happens is when you when it becomes active, it will activate a G protein, alpha S, alpha I, alpha Q, any one of those G proteins. And when that happens, the alpha separates from the beta gamma. The beta gamma then will attract this this kinase here. Let's zoom in on this. This isn't the best picture. It's kind of busy yesterday and I didn't do as good a job as I should have. Okay. Okay, so here's the enzyme that's going to be activated by the alpha. So that gets activated, could be adenyl cyclase or something, but that always leaves your beta gamma hanging out, and it's got a lipid attachment. So it's going to hang out near the membrane. When it becomes free, then it provides an attachment point for GRK to come up to the membrane. Recruitment, we talk about that a lot in cell biology. So when GRK gets recruited to the membrane, it then phosphorylates the G protein coupled receptor in those sites on the tail. Here's the hormone still attached. Now, once it does that, the protein beta arrestin will come up and bind to that tail. Now, if you remember, this loop here between 5 and 6 is interacting with G proteins and turning them on. So when beta arrestin comes up, it blocks that. So at this point, this receptor still got hormone attached, still in the active conformation, but it can't interact with G proteins anymore. So it's effectively desensitized. And also this beta arrestin will bind to a protein like clathrin, and then the whole receptor beta arrestin hormone complex will come into one of these clathrin coated pits. Relax you. Okay, so now we've got the receptor in the clathrin coated pits. This AP2 is just an adapter protein that becomes part of the complex. The whole thing then invaginates. Dynamin comes in and seals off that endocytosing vesicle and the vesicle endocytosis. Once it's endocytosed, the clathrin, dynamin, and AP2 come off and they get recycled back to be used again. And then you've got the receptors in that light vesicle pool underneath the membrane. Now these, several different things can happen with these. They could just kind of hang out here and be in that pool that goes into and out of the membrane when the cell needs it, or they could make their way over to some primary endosomes, attached to primary endosomes. This gets acidified, the hormone comes off, and the receptors can then be put back into the membrane here, or they may go right to become that endosome might become a lysosome and then the receptor destroyed. So at all times you want to think that there are receptors in the membrane, there are receptors in this light vesicle pool, and there are vesicles being made and degraded. So you've got 4 different processes going on at the same time. You've got endocytosis, bringing them in, exocytosis, putting them back, and then making new ones and destroying old ones. So how many receptors are in the membrane is based on homeostasis between those 4 different processes. But mainly it's between endocytosis and then reinserting to the membrane. So short-term response to agonist stimulation then is to bring them in. When the agonist goes away, they go back out. And then longer-term chronic changes involve destruction of the receptor, or if you're using an antagonist, production of new receptors. Okay? How does this affect our patient? We've got patients in this class. So here is the difference between receptor binding to beta-1 receptors in the heart in normal versus failing hearts. So we haven't talked about this yet. This will be an important part of the class later, but congestive heart failure is when the heart is not beating enough to pass enough blood to meet the needs of the body. So the body compensates by making it pump harder and faster, which is beta agonist stimulation, right? But chronically, over time, if you have to keep stimulating the heart to make it beat faster, you start to downregulate those receptors and your compensatory mechanisms are not as effective as they used to be. So we can see this here at the receptor level. This is your binding isotherm. This straight line down here is the nonspecific binding. The upper one is the total binding, and these 2 curves should be linear at this point. And then here is, like I said before, you subtract the nonspecific from the total, and this is your specific binding. So from this specific binding curve, you find your Bmax. Here's the scattered plot, which is just the linearization of this model, and you find the Bmax by the x-intercept here. So this is a non-failing heart, normal heart. It's got about 9 femtomole per milligram protein of receptors, okay? Femtomole, what is it? Milli, micro, nano, femto, atto. So femto is below micro. No, micro, nano, femto. So femto is below nano. So it's small. You don't expect a lot of receptors in a cell. So anyway, 9 femtomole per milligram. Here's your failing heart. It's got about half as many receptors, 45. So you've got downregulated receptors. So this is from a paper in I think the late 80s, early 90s or something. So the idea was you have a hard time, when someone's in congestive heart failure, you have a hard time treating them with beta agonists to stimulate the heart to go stronger because the receptors have already been downregulated. And I saw some papers that said, again, from the same time period, that if you treat your patient with beta-1 antagonists, the receptors come back up, which is good, but they're blocked now. So what good is that going to do? I don't know. That's neither here nor there. So here is a different paper by the same people. We're looking at total beta receptors in beta-1. So beta-1 are the cardiac specific ones. Beta-2 are respiratory. They're in the bronchioles. So total number of beta-1s here in non-failing, left ventricle versus right ventricle, and in the failing, left ventricle versus right ventricle. So you can say 68. Left ventricle is the most important one. That's the systemic circuit. So 68 versus 32. So it's basically in half. So receptor downregulation is really important in this disease process. And here's the difference in functional agonists. This is your dose response curve. So this is increase in milligrams of tension. So how much stronger does it get when you add your beta agonist down here? In a normal heart, you get a good gain. We call this gain of function. And in the failing heart, a lot less so because it doesn't have as many beta receptors. Okay, so there's functional consequences to the lack of receptors there. Okay. 15 minutes. That's all I had to talk about today. Yeah, I figured it wouldn't take long, but we could spend some time doing some review so that, you know, on Thursday we'll all be ready for the test. So I posted a test online. Let me pull that up now so we can all look at it. It's in your Moodle page. It's called Pharm Test 110. So this is the exam I gave. This is the exam I gave in 2010. Sharon is Karen. Oh, there's a chat. Can we do the graphs on the practice exam? Can we do the graphs? Not sure what you mean. Oh, I haven't looked at the, can we, oh. Yeah, I don't know what you mean. Sorry. Can we do the graphs? What do you mean? Like, can you explain them? You know, if I could explain them, I would, but frankly, I'm as confused as you are. Kidding, kidding. Let's look at this practice exam. And if this is what you're talking about, we'll talk about it. But if not, we'll just ask me again. We'll figure it out later, okay? Okay. Oh, I really don't like this Google Word doc or whatever. I don't know how to use it. Let's blow it up. Okay. All right. Ooh, there's a kitty in the back. I like that. Celine's got a kitty in the back there. Okay. I'm going to leave Celine alone because she's playing with the cat. So Mike West, answer the first question for me. In molecular terms, what determines the affinity of a ligand for the receptor?
Is it just the shape of the molecule, like the ligand and the receptor? That's it?
Yeah, but I want you to go in more detail about it. Like what about the ligand and what about the receptor? I think I talked about this in the first class. Why would one ligand be better than another? How do you make new drugs? I can move on. Anyone want to help?
Test a lot of molecules, see what's similar to what and what's more effective, and then try and synthesize something better.
Right. How do you get different molecules? What makes different molecules different?
Functional groups,
okay,
and the way they're formed, whatnot.
Functional groups, right. And why do the functional groups change the affinity?
Because covalent bonds are different lengths and different strengths, and then when they bind, they pull on the receptor and make that conformational change.
Right, right. So what I'm trying to get at here is you've got functional groups on the drug, on the ligand, you've got functional groups on the amino acids of the receptor, and they're interacting with each other non-covalently, and that's pulling the receptor and changing its shape, its conformation, right?
Yes.
Okay. When I say right, just say yeah. Of course you're right. You're Dr. Slish, you would never lie. What is the difference between KD and ED50? How is each determined? When would they be different? Ooh, this sounds like a question for Noemi Alonso. Yeah. Thought you were gonna have a quiet morning. You're wrong.
The KD is like a measure of the affinity of the drug. So it measures like the attraction between the ligands and the receptor. And you measure it experimentally, but I forgot the equation.
That's okay. That's good enough. I won't expect you to know the equation.
Is that what you were asking for? I think is. So what were you asking when you said how each is determined?
Okay, so how do you find the KD? You said
experimentally. Is that what you were asking for?
Right. But like, what would you call those experiments? What are those experiments called? Remember?
Binding isotope?
Radioligand binding?
Oh, yeah.
Okay. So ED50. How might one?
Effective dose?
Uh-huh.
Yeah.
And how do you find that?
Don't remember. Hold on. You measure the potency, the concentration needed to produce the effect.
Right. Dose response curve, finding the potency, and that's the ED50. Okay, good. Let me bother someone else real quick for the last question. Celine Ramos, when would KD and ED50 be different? Muy importante. I had a whole day about this. When is KD? I'll give you a hint. Furchgott, rabbit arteries.
I don't know if that's a good hint.
Well, if I had some spare time, maybe I'd tell you about
Oh,
it.
because the amount of spare receptors
Right, exactly.
involved?
Exactly. If there are no spare receptors, then KD and ED50 are the same. But if there are spare receptors, then your ED50 can be a lot lower than your KD because you don't have to activate all the receptors to get response. Good answer, Dr. Slish. Okay. I've been studying this all night, so I know this stuff cold. Discuss or diagram the system of like cyclic mediated protein K. Okay, so I'm not gonna do that now because basically it's a drawing. You want to draw the receptor activating GS and GS breaking into alpha S and beta gamma, activating adenyl cyclase, and cyclic AMP activating protein kinase A. So, ooh, let's discuss this. This was just today. Ariana Totaram, you look bright and interested in my lecture today. Can you, off the top of your head, tell me about the GRKs?
Doesn't it have to do with it binding to an arrestin and then the arrestin binds to the calthurin or something?
Almost. Arrestin's part of this. How does GRK get activated, and what does it do? Why don't we move on to Matt Bernhardt? He thinks he's smart. I think he's smart. I don't know. Matt, you're muted.
Yeah, no, my thing was minimized or whatever, so I couldn't figure out how to get there. Yeah, I'm not too sure about this one.
Okay, so we just talked about this today, so I don't expect you to have that memorized yet. But basically, unless you learned it in a different class, the receptor activates the G protein. The beta gamma that's left behind then brings the GRK up to the receptor, and it phosphorylates the receptor. That phosphorylated receptor then becomes a target for the beta arrestin to come up and bind to it, and that causes the receptor to go into the coated pit and be internalized. So that's the GRK thing, but we just did that today. Okay, here comes one of the hard questions. And just in time for Bizawit to answer. Okay, focus Bizawit, this is all on you. A new drug, OPC 593, increases PIP2 turnover and calcium cycling in a liver cell line, HEK cells. what are 3 possible mechanisms of action? How about you give me one? So you got this drug, and all you know about it is when you treat liver cells with this drug, you get breakdown of PIP2 and calcium release. So what's one possible target for this drug? I like questions like this because there are a lot of answers, and you just have to get past the understanding of how drugs work. And once you know how drugs work, then the answers start to come out. So basically, the drug's going to bind to some protein in the liver cell and cause PIP turnover and calcium cycling. So what's one potential target for this drug? Ron Bizawet, right? You're muted.
Calcium receptor?
I'm sorry, say it again.
Maybe the calcium receptor?
Calcium
Okay.
receptor. Which calcium receptor? The only calcium receptor I know is the ryanodine receptor, and that's not going to be part of this. So what, Dante Quinones, what's one target for this drug? Well, what normally causes PIP turnover and calcium release? Okay, everyone see, does anyone know, or should I stop and, does anyone know this?
Isn't it like, isn't it since like you're having more calcium, there could be more calcium channels being opened up?
Not really,
No.
no. PIP turnover. So
You're looking for parts of the mechanism that the drug affects. It's like that's like just like a receptor or a molecule or something.
exactly.
Okay, one idea, maybe the alpha subunit has less GTPase activity. It makes the alpha subunit have less GTPase activity, so it never comes off of the AC. And
Okay, excellent. Which alpha subunit or which alpha subtype do you remember which one activates phospholipase C?
is it alpha S or alpha Q?
Q, alpha
Okay.
Q. So that's one potential target. It could be binding to alpha Q and activating it by blocking its GTPase, and then it just stays active and is increasing PIP turnover. Okay, good answer. How about another answer? You can go upstream or downstream of alpha Q, and either of those proteins is a good answer. Who do we got? Everyone's quiet today. Do I have to start having coffee delivered to each of your dorm rooms? I'll do it. I had my coffee. Okay, what's upstream of the alpha Q? What activates alpha Q? Matt Bernhardt. You're muted again.
I know I can't find my Zoom. I'm like trying to like look through the notes at the same time as this.
Oh, okay.
Um,
This is lecture 3, I believe, or 4. Lecture 4.
all right, let's check out lecture 4.
Ariana. Ariana.
Maybe cyclic AMP.
No, that's not part of this system. Cyclic AMP is its own system. Someone said something on chat, but I can't bring it up for some reason. Let me stop share for a second so I can bring my chat up. Phospholipase C. Okay, good. Noemi. So this is, oh, I've been watching my chat. So a phospholipase C would be downstream of it, but yeah, that would be an answer. So this drug could be directly activating phospholipase C and causing PIP2 breakdown. Good. Okay, okay, so there's one more answer. What's upstream of the G protein and phospholipase C? So going backwards, phospholipase C, GQ. What activates GQ? Mike, you said it earlier.
The norepinephrine receptor.
The receptor, right. It doesn't have to be norepinephrine, but some receptor would be activating this GQ. Okay, so let me just review. It's like, this is the kind of question I love to ask because it's kind of like broadens the way you think about things. Like a drug can bind to any protein in the system and activate it or block it. And that's the way drugs work. So if you look at the cyclic AMP generating system, you've got your beta adrenergic receptor, right? You can activate that with a drug, like isoproterenol can activate it. So that's one place of drug action. Then you've got GS. That can be activated by pertussis toxin, will directly activate that. Then you've got adenyl cyclase. That can be directly activated by a drug called forskolin. So like everywhere along the way, a drug can interact with the system and turn it on. And that's kind of a lot of what pharmacology is sometimes. When you have a drug, you get a response. How do you connect those two things? And all the experiments you do to find out where exactly it's working. Okay. Question 6. I'm going to read this. You can pull it up from your Moodle page if you want. I'm not going to share because I'm that kind of guy. The first paper we read discusses changes in the dihydropyridine receptor in disease state. What did they find and how did they relate this to disease? Ooh, this expects you to have read that paper, or at least we're in that class. So remember the paper, dihydropyridine receptors. How do they change in disease state and how does that affect the function? Everyone's waiting for someone else. Should I call on someone? Oh, let's do this. Let's everyone chat and nominate someone to answer this question. And it's on you. Okay. Ariana, who should answer this question? Either you answer it or you nominate someone. Ha, double jeopardy.
Nominate you.
No. I don't know the answer anyway. I mean, I read that last week. I've already forgotten what was in the paper. I would never lie to you. Boy,
Don't the receptors like downregulate when the disease state's like further down the line when it's more chronic?
Do you remember the disease state? What was the disease?
No.
It was created by cecal ligation and puncture. You remember this?
Was it sepsis?
Sepsis, right. So bacterial infection in the blood. And what happened to the receptors early versus late?
There was more receptors at the beginning, and then once it got chronic, there was less receptors.
Right. And how did that correlate with the function? It's going to be an open book test, so you don't really have to know anything in the end, I guess. Teasing you, Celine.
Mm.
Okay, so dihydropyridine receptors are voltage-dependent calcium channels. So early in the disease state, they go up in the membrane and the heart beats stronger, excuse me, as you would expect with more calcium receptors. Chronically, once you get into the chronic disease state, then they start to be removed from the cell surface and the heart beats less strongly and then you die. Okay, everyone remember that or no? Just say yes, yes, Dr. Such, remember that. Make sure you review that because I will have a question on the exam about that, something about that paper. So make sure you look that over.
They affect the contraction of the heart, right?
Right, right. Because if you remember the contraction of the heart, it starts with calcium coming in through that calcium channel, and that gives you the sloped action potential. It goes up, and then you've got that plateau phase, and then it comes down. So that plateau phase is calcium coming in through the calcium channel while potassium is going out through the potassium channel. So that gives you a plateau. And that calcium coming in is the trigger calcium that activates calcium release from the ryanodine receptor to give you contraction of the cell. Okay. All right. So if you have fewer of those receptors in the membrane, you've got less plateau, less trigger calcium, weaker contraction. Okay. Number 7, I think. In the following experiment, so in the experiment you'll skip over this one because it requires you to look at a graph and there's no graph in the paper, so we can't really answer the question. All right, let's move on to number 8. Ooh, I'm going to share this one because this is pretty complicated, so I want you to be able to see it while we talk about it. Sharon is Karen. Okay. And this is about what we talked about today with GRK. So remember the whole GRK system. Receptor activates G protein, beta, the beta gamma subunit brings GRK up, GRK phosphorylates the receptor, arrestin comes in. Okay. In an experiment testing downregulation of receptors, you do an overexpression experiment with a catalytically dead GRK. So basically, you're putting a gene into these cells that is going to like flood the cell with GRK, but GRK isn't active. It's got no kinase activity. This is a protein that's structurally identical to normal GRK, but with one amino acid change, so it cannot phosphorylate targets. Okay. After you check to make sure that this protein is expressed in the cells, so you know it's there, you stimulate them for 2 hours with dopamine. After this pre-exposure period, you measure receptor numbers in normal cells, control, normal pre-exposed cells, GRK2 catalytically dead cells, not pre-exposed, and GRK cells pre-exposed to dopamine. Here are the results. Okay, so you need another graph here, which is not available. So let's just talk about this. You've got some controls, you've got 3 controls and 1 test. Let's just talk about what these controls are doing. I'm going to go slow because I know this is complicated. Okay, so you're taking a catalytically dead form of GRK and injecting into these cells. What do you think that's going to do? Keep in mind, our experiment here is to expose cells to dopamine for a long period of time and then measure the number of receptors. What are we measuring here? Silence. Why might you do this experiment where you pre-expose for a while and then count the number of receptors? What are we looking for? Pre-exposure, and how's that going to change the number of receptors? Oops, that was the answer. Who haven't I bothered in a while? Ariana. Sorry. What happens to receptors if you expose them to agonists over a long period of time?
Wouldn't you be looking for potency and efficacy?
Not in this particular experiment. We're just looking at binding here, numbers of receptors. But what normally happens with the numbers of receptors if you overexpose with an agonist for a while?
It usually decrease.
Right, downregulation. So what we're looking at here is downregulation. So you've got 4 experiments going on. Number 1 is just control. You're just going to expose to dopamine for 2 hours, measure the number of dopamine receptors. Wait. Let me make sure after the exposure period. Okay, so no. So your first one is just taking normal cells that haven't been pre-exposed, measure the number of dopamine receptors. That's our baseline, okay? Then you're going to take a second set of cells, expose them to dopamine for 2 hours, wash the dopamine away, measure the number of dopamine receptors. What do you expect to see, Matt?
I mean, you just said downregulation, but I have the graph on mine, and it looks like it increases receptor binding.
You're lying. You're lying to me. Can you
share my? Yeah, I can share my screen.
Oh, cool. Do that.
Yeah. How do you do this? Share screen.
I don't know if you can. I think only the person who's in charge can share.
It's
Why don't
only
you
the
do
host
this?
can.
Can you copy it and email it to me, and then I'll share it?
I'll just send the whole Google Doc or the Word document.
Okay. So what the hell was I talking about? Okay, so let's just do this first experiment first. So control, normal cells, how many dopamine receptors? Then you take similar cells, treat them with dopamine for 2 hours, wash the dopamine out, measure the number of dopamine receptors. What do you expect to see?
Is this
Nobody's
question for
answering.
me still? Oh.
Well, sure, if you got it. I'm just throwing it out to the crowd there.
Nah, I wasn't listening. I was trying to focus on emailing.
Okay, you do that. I can't see my chat window. Let me stop sharing for a second. The numbers of receptors increase. You're pre-exposing with the agonist for 2 hours. Do you expect the numbers to increase or decrease?
I just sent it to you.
Okay. What do you say, Bizu? If there is downregulation, wouldn't the binding of receptors increase? No. If there's downregulation, there'd be fewer receptors. So decrease, right? Noemi says decrease. So the idea here is if you take this tissue, you're pre-exposed with dopamine for 2 hours, the tissue gets used to that. I hate using that term. The tissue adapts to that high level of activity, pulls the receptors away from the membrane. You wash it away and then test it. There should be fewer there. Okay, let me get this email.
I think I just read the graph wrong. You were right the whole time.
You'll get used to that, Matt, me being right. Okay. Damn. Oh, good. All right, so I can see it now. Okay. So let me share this. Thanks, Matt. Thanks for bringing that up. Okay, so these little control is these diamonds here. So that's what we're testing everything against is those diamonds, okay? So that's your control. And then your control pre-exposed is this one, and you're here. So you went from here to here in your binding. So that shows you've got fewer receptors, okay? So downregulation. Now, now the second part of the question, oops, shit, son of a bitch. After this pre-exposure, you measure the number of receptors in normal cells. Normal cells pre-exposed to dopamine. GRK cells that are not pre-exposed. That would be your GRK control. Okay, so this GRK control is up here. All right, so these are cells that you've put this catalytically dead GRK in, and then test how many receptors are there. Is there any difference from control? This is no. Everyone say no. Noemi, you okay? No, okay. So this is control, and this is GRK dead plus control. All right, so now you're going to do pre-exposure of these dead GRK2 cells with dopamine for 2 hours. Take the dopamine away, measure a number of dopamine receptors, and you get this. All right, what does that tell you? Two different things. Oh, come on. Two different things this tells you. And I will give $100 to anyone who can come up with one of them. Yeah, I went there. This is my bald head glowing. $100, people. You're college students, you got to be poor. It's the law. So was there downregulation? Question one, with the GRK dead cells. Dante, cut me a break here, Dante. You know this. Is this really hard? You guys are not, or you guys just don't want to talk? Okay, let me go through it again. Well, I'm sorry, go ahead.
For the GRK2 control, there's dead GRK, right? Something like that.
Yep, dead GRK, but not pre-exposed. Just how many receptors are there? Comes out the same as control.
So there's a normal amount, correct? Like it doesn't change.
Right.
It depends if they're pre-exposed to it, because that causes downregulation.
And then when you pre-expose, it causes downregulation. Right, good. So we're still getting downregulation, even though we've got this enzyme in there that's going to block basically all GRK activity. So, but we still get downregulation. What does that tell you about the downregulation?
There's more mechanisms to it than just the GRK.
Exactly, exactly. You can see it's important, right? Because if you have GRK, you get this much downregulation. Without GRK, you get this much downregulation. But it's important, but it's not the only mechanism there. There's something else going on causing downregulation.
Okay.
Okay, does that make sense? Let me just go through it all again, just to make sure it's clear. So basically what I'm doing here is saying it's an experiment to see what is the contribution of GRK to the downregulation of receptors. So you've got these cells that have got GRK in it, and you measure the number of dopamine receptors. Okay, that's your control, that's your baseline. And then you take those cells and you expose them to dopamine for 2 hours, and you find out how much downregulation happens in 2 hours with exposure to dopamine. And you see, boom, quite a bit. It's like half as many receptors, basically. All right, so then you take a different set of cells and you put this GRK catalytically dead subunit in there, which is basically going to wipe out all GRK activity in the cells. So there will be no downregulation due to GRK. And you test those cells and they come out same as control, okay, which shows you that that catalytically dead didn't affect the control level of receptors in the cell. Okay, so now we're ready to do our actual experiment after 3 controls. And when we pre-expose the cells to dopamine for 2 hours and then measure the number of receptors again, we still get downregulation, but not nearly as much as we did before. Okay, now we're down to, probably a third fewer receptors instead of half as many receptors. Okay, so that's showing us, number one, GRK is important here because this much downregulation from this point to this point is probably due to GRK, because when you wipe out GRK, you no longer have that much downregulation. And number two, we learned that there are other mechanisms there. We talked earlier about protein kinase A phosphorylating the receptor too. So you've got other mechanisms that are downregulating the receptor as well. Okay. How many people think this is complicated? You can put your hand up. Yes, yes. Okay. This is probably the hardest question I would ever ask. This is a hard one. And I think if you just knew that it had something to do with downregulation, that's 5 out of 10. And you start talking about GRK being important for downregulation, that's 7 out of 10. You get all of the whole schmear is worth 10 out of 10. So that is realistically a hard question. Okay. All right. Any questions about the exam? Do you want to practice some more? Shall I pull up another old exam? Yes? This is yes. This is no. You guys are so quiet. Jesus. You're home. Nobody can see you but me. It's okay.
Can you do number 7 now, since you have the graph for it?
Okay, I can do that. Oops. Hang on a second. Share screen. Number 7 is similar, I think. It also has to do with downregulation, I think. Oh, this is a good question. These are the ones that I write and I go, okay. In the following experiment, aortic smooth muscle was exposed to norepinephrine for 1, 2, and 5 hours. After the time of exposure, norepinephrine was washed out and a dose-response curve was constructed after the contraction of the tissue with norepinephrine and compared to control tissue not previously exposed to norepinephrine. What conclusions do you make? Okay, so this curve is your normal dose response of contraction with norepinephrine. And then you take that tissue, you expose it to norepinephrine for an hour, and then wash the norepinephrine out and let the tissue, recover for a few minutes. And then you do a dose response curve again, and you see this decrease in potency. And then you take a different tissue, you pre-expose it for 2 hours and wash it out. And then here's your dose response, further decrease in potency. And then you expose the tissue for 5 hours to norepinephrine. And then now you're going to decrease in effect. What conclusions would you make based on this? If I were to give you a hint, which I won't, but I will, what does this series of curves remind you of that we talked about? Basically, I'm taking two different concepts from the class and putting them together here. What do these dose response curves remind you of?
Spare receptors?
Spare receptors, right. If you've got spare receptors and you treat with an irreversible antagonist at higher and higher concentration, you see curves like this, right? Okay, back to this. Now we're talking about pre-exposure with norepinephrine. So how do you combine that idea with the spare receptor idea?
Maybe there's still spare receptors up until a certain point, and then after, well, if somewhere between 2 and 5, there's the amount of receptors is decreasing to the point where there aren't any spare ones left because you can't reach the full efficacy.
Right, exactly. And why are the receptors going away?
They're being downregulated because there's so much norepinephrine.
Exactly, exactly. So that's what I was getting at here. With your pre-exposure, you're downregulating receptors, but in this tissue, you've got spare receptors. So instead of seeing a decrease in effect, you're seeing a decrease in potency up to a point where, like Mike says, you've got too few receptors. Now you're seeing a decrease in effect. Okay, so that's a classic Slish question too, where you take two different concepts and you put them together.
I have a question.
Okay.
Does those three graphs have the same KD or no?
These three.
The first three graphs, yeah.
The first three have got a different KD. Or no, KD. I'm sorry. No, KD is going to be the same all the way through because it's still norepinephrine binding to the same receptor. So KD is the same all the way through. The only way KD would change would be if we use different agonists instead of just norepinephrine every time. Okay. Is that it? Shall I bring up another exam? We've got time to kill. Yeah, let's do that. Let's reach into the vault. How many people were born in the year 2000? One. This is the first year I taught this class. Let's see what this exam is like. It could be totally different. Oh, that's not an exam, sorry. Tests. Year 2000. I'll show you how much life really changes for a professor. Two important terms are KD and Bmax. Define these terms. Pretty much the same as 2010, okay. Two different receptor types that respond to acetylcholine are muscarinic and nicotinic. All right, we haven't talked about this yet. Latch state, we haven't talked about that. Describe or draw events in postsynaptic cell where alpha, okay. Again, this is just a memorization question. What happens when an alpha receptor is activated by norepinephrine? And that's just the IP3 calcium system. And that's something we haven't done yet. All right, we haven't done this yet. Yeah, I'm doing things in different orders, so none of these questions really apply. Oh, maybe this does. Recent studies have shown that overexposure of arteries to norepinephrine results in the synthesis of an inducible form of nitric oxide synthase in smooth muscle cells. This enzyme produces nitric oxide inside the cells. What would be the result of this enzyme induction? Okay. So you've got arteries, you expose them to norepinephrine, and you get an upregulation of the protein nitric oxide synthase. What does nitric oxide synthase do?
Doesn't it?
Sorry, go ahead.
It turns arginine into nitric oxide so it can send it through the gap junction, right? And then stimulate the next cell?
Not gap junction. It'll just diffuse right out of one cell into the other cell.
Okay.
Okay, but this is already inside the smooth muscle cell. So Ariana, what does nitric oxide do in cells?
Isn't it a regulator that produces like relaxation?
Mm-hmm. Yep. Do you remember the second messenger? Nitric oxide causes the production of cyclic.
AMP?
No. That was a trap. That was a trap. Cyclic what?
GMP.
GMP. Excellent. So nitric oxide produces cyclic GMP that causes relaxation. Okay, so the question was, what would be the result of this enzyme induction? So you're exposing to norepinephrine causing contraction, and this protein comes up and starts to produce nitric oxide. What's going to happen?
The muscle will be more prone to relaxation. They just, yeah.
Right, exactly. Physiological antagonism. This nitric oxide is going to cause relaxation with the norepinephrine there. Why do you think this mechanism exists in smooth cells, Dr. Ramos?
Is it so they don't get fatigued, so they can like reset everything?
Probably, yeah. It's like a negative feedback cycle, basically. Negative feedback is all. Okay, that was a good question. That was from 20 years ago. I was a smart guy back then. Oops. Okay, let's try another one. Try a more recent one. Watch, I'm going to end up giving you the ones that are on the exam already, because that's the way I roll. Thumbs up there. Okay, I'll remember that. Okay, here's it. Okay, I'm not going to go over the first questions because they're just memorization, so I don't want to have to waste time with that. But question one is, what information can you learn from Bmax and Kd determined in radioligand bindings? So I talked about this, I think, at the end of lecture 1 or the beginning of lecture 2. What can you learn from binding experiments? So you can look that up again if you want. Boy, this question keeps coming up, doesn't it? Okay. Hint, hint. Discuss or diagram of cyclic AMP production. Okay. Diagram, downregulation by GRKs. See, it's the same stuff every exam. It's called being lazy. OPC increases PIP2 turnover. Okay, we talked about that one earlier today. Here's this question again. Jesus Christ, I recycle all my questions. You know, every exam I try and come up with at least 1 or 2 new questions, but after 20 years of teaching, it's like I've got a ton of questions, so I just add a couple more each time. So I do recycle a lot. Okay, you, Matt Bernhardt, are a big hotshot pharmacologist at Merck Pharmaceutical, and the medicinal chemist gives you a chemical that she made by adding functional groups to an agonist of a alpha-1b adrenergic receptor. So you've got a drug that's a specific alpha-1b agonist, and you add a couple chemical groups to it. You want to determine what the effects of this new compound are, i.e., is it an agonist, antagonist, partial agonist? What type of subtype of receptor acts? So you use your model of resistance arteries, rabbit ear artery contraction, to determine its effects. What experiments do you do? I don't know if we didn't really talk much experimentally, did we?
I mean, you might be able to do the same one they did for the GRK is like catalytically dead or whatever, and you can see if it's an agonist or not, because if it downregulates the receptors, maybe.
See if it downregulates the receptors. That would determine if it's an agonist. That's true. What's another way to see if it's an agonist, though? Maybe an easier way than that experiment. How do you know if a drug is an agonist in this tissue for contraction?
Isn't it if it's producing the same contraction, then it's an agonist?
Yep. If it produces the same, if you add it and it causes contraction, it's going to be an agonist of some sort. Okay, how do you know it's an agonist of the alpha-1b receptor, though? What might you do to check that? Like, make sure it isn't activating a histamine receptor or something bizarre. How do you know?
Can you do an SDS page and then just like isolate the adrenergic receptor and see if that binds to that specifically?
That's interesting. Okay, so isolate that receptor and see if it's binding specifically to it. That's a good idea. Maybe a less complicated pharmacological way, which maybe we haven't covered enough pharmacology yet, but a pharmacological way would be if you had an antagonist for the alpha-1b receptor and add that and see if this new drug still produces contraction. If it does, then it's using a different receptor. But if it doesn't anymore, then you know it's the same receptor. How could you tell? Gotta make sure I don't have a tick here. I'm in Pennsylvania, gotta watch for ticks. How can you tell if it's a full agonist or a partial agonist?
It's a partial agonist because you're only getting some contraction, but not the full contraction that's going on.
That's a good answer, but it's not all the way there, because remember, we're talking about rabbit ear artery that has a lot of spare receptors. So if you didn't have spare receptors, that would definitely tell the tale. But what else do you want to do with this to see if it is a full versus partial here? We've kind of discussed this already today. How do you know there's spare receptors there? Because you said there were, Dr. Slish. Dr. Slish never lies. That's not true.
Because down regulation didn't mess with the ED50? No, it did mess with the ED50. The affinity, like the same curve, the KD.
Right. That would be one way of looking at it. Yeah, definitely. If you start down regulating receptors, how does it affect the KD or the Emax? But backing up, forgetting about this new drug, how did we figure out to begin with that there were spare receptors in a tissue? Going back to our friend Mr. Furchgott. Like I know him by now. So going back to Furchgott, how did he know that there were spare receptors in his rabbit ear arteries? Emax, KD, right? Emax are separated from KD because you can get full response with only activating 6% of the receptors. So that's something else you might want to do here with this new compound. Find its KD, find its ED50. If it's a partial agonist, I'm lost now. I started thinking about something else while I was explaining this, and I was like, that's a more interesting thought than what I'm doing right now. This COVID stuff, this isolation is it's hard on concentration abilities. Okay, what were we talking about? So how do you know if it's a full agonist or a partial agonist? Okay, so you're getting full response, but are you, yeah, I lost my thread. Let's just forget about this one. My brain is fried at this point. 10:31. Okay, any other questions? Comments? Stopping the share. Can you go over 7? When was this? Oh, that was 15 minutes ago. Do you still want me to go over number 7? Noemi? No?
You already did.
Already did. Oh, okay,
Yeah.
okay. Okay. Oh, that's the one Matt said go back and do 7. Okay.
Yeah.
All right. Any questions, comments? Shall we call it a day? So your exam, I came up with, it's all in one thing, but like the first 10 questions are like multiple choice or drag and drop. Some of the drag and drops have more than one answer per. They have like 4 or 5 things you have to drag and drop. So those questions are worth 10 points. Then the multiple choice are worth 5 points apiece. So the first 10 questions or first 7 questions together or something are worth 50 points. So all that kind of objective stuff is 50 points. Then there's 5 more questions worth 10 points apiece that are essay questions, like the ones we did today. So the first 10 shouldn't, or the first 50% of the test shouldn't take you that long. It's pretty plug and play. And then the second ones you want to spend more time on. So I figure an hour and 15 minutes for this, like if you were in class, that should probably be enough time. Okay, everybody okay with that? Yes? Okay, any questions about the test or should we just call it a day? Going once, going twice. Okay, I'm gonna say goodbye. Goodbye, people. So how about on Thursday, we meet on this and then I'm gonna we meet on this just to say hello and make sure everybody is up and alert. Then we'll start the exam and I'll shut the Zoom off. And then I'll be waiting. If you have questions, you can email me online, okay? All right, we'll see you Thursday, guys. Have a great rest of your week.